Namaskar दोस्तों, welcome to channel Toxicology on board. In this video, we will be learning about another DABT topic, that is Principles of Toxicology, Clayson Chapter 2. In this video, we'll discuss question number 6 to 10 from this Clayson short textbook is there, no? So previous video had question number 1 to 5. Here we'll discuss question number 6 to 10. Let's directly dive into the question. The question is, when a chemical A and B are administered simultaneously, their combined effects are far greater than the sum of their effects when given alone. Okay, A plus B, they're mixing. The elimination interaction between chemical A and B can be described as which of the following? So basically, from this question, we need to learn about basic concept, that is potentiation effect, additive effect, antagonistic effect, functional antagonistic. The right answer will be in the green text, that is synergistic effect. A plus B more than individual A action as well as individual B action. Okay, let's see what is the explanation which is given in the Clayson 9th edition, page number 31. Synergistic effect is observed when the combined responses of two chemicals, in this case chemical A and chemical B, are much greater than the sum of the responses of each chemical given alone. 2 plus 2 is equal to 20. Example, carbon tetrachloride, ethanol. They are both are causing liver toxicity. Alcohol is causing liver toxicity. Carbon tetrachloride, which is used as an or as a solvent, industrial solvent, it is also causing liver toxicity. But when they are the exposure, when A plus B is there, both carbon tetrachloride and ethanol, together they produce much more liver injury than expected based on the extent of damage at given dose when administered alone. So what we need to learn in more is that in you read in page number 31 Clayson textbook only 9th edition, figure 2.1 is given. So let's discuss this particular figure. See, this 2.1 is nothing but it's an illustration of responses following various chemical interactions. Okay, A plus B plus C interactions. Data illustrates a theoretical response to these three different chemicals, chemical A, chemical B, chemical C, either alone or in combination. As shown, chemicals A and B have measurable responses when used alone, whereas chemical C does not. C is not having any response. You can see individual chemicals A, B, and C, the response scale is there. A is showing some effect, B is also showing some effect, C, no effect. Four different examples of chemical interactions. We have A plus B, A plus B, A plus C. Wow, the different we'll see. Namely, additive. A plus B is additive. A ka effect is this much blue and B effect is red color. Together A plus B is equal to same only. That is why it is called additive. A plus B addition is taking place. 2 plus 2 like 4. Coming to synergism, A is there, B is there, but A plus B, 2 plus 2 is not really resulting into 4, but it will result into, for example, 5 or 10 like that. Coming to third one is potentiation. Here in this case, A is combined with the chemical C. C as such alone when administered it has no effect, but when it is combined along with the drug A, chemical A, there is a potentiation of effect. 2 plus some 3, it will end up with 8 or 10 or 12 like that. Coming to antagonism, antagonism is very important principle which is used as an antidote example in the case of toxicology antidote. If you want to inhibit certain effect of a certain chemical, then you have to use antagonism principle is important. So in this case, chemical A is there, chemical B is there. What it is doing? Overall effect A plus B, you can see it has been decreased. Here the effect is antagonism. Exam point of view, what you will get? They will give you examples and they will ask you to identify the option. Definitely. In this case example, we have seen CCl4 as well as ethanol. But try to read other examples. Maybe in one of my videos, I will try to list out all the examples also. Okay. Next one is question number 7. That is with respect to dose response relationship, which of the following is true? We need to identify the true statement. Graded dose response relationships are often referred to as all or nothing responses. Actually, all or none is present, either an effect is present or not present, that comes under quantal dose response. It is not graded dose response. Next, quantal dose response relationship allows for the analysis of population response to various dosages. Very important in terms of epidemiology also. So population of individuals can be done by quantal dose response. Quantal relationship characterize the response of an individual to varying dosage. Quantal that is graded response actually. Quantal dose response describes the response of an individual organism to varying dose of a chemical. Again, this is a as dose is related, it is graded response. Next, the dose response always increases as the dosage is increased. No, this is not a. There are certain times saturation will be there. So this statement is also not right. So right answer will be in the green text, that is quantal dose response relationship allows for the analysis of population response to various dosages. So we'll also try to compare the graded dose response, quantal dose response, and each feature under response. If you see graded dose response, it is a continuous scale. Examples we have, units we have millimeter Hg, percentage, contraction, etc. Whereas muscle contraction. And coming to quantal dose response, either it is present or it is absent. Coming to subjective feature, this graded dose response, single biological unit, individual or organ response you are seeing. Here, population of individual. That's why this our answer will match with this particular statement. Coming to y-axis measure, if you see dose response curve, intensity or magnitude will be given on the y-axis. Here, frequency of effect in the population. How frequently, how probability, like that, those will be measured on the y-axis in the case of quantal dose response curve. Key metrics. Here you are seeing potency EC50 and efficacy Emax in the case of graded dose response, whereas in the case of quantal dose response, ED50, TD50, LD50, and therapeutic index are seen. Coming to main use, graded dose response characterized drug action and maximum effect will be there. In the quantal dose response, assessing population variability and safety margin will come under this quantal dose response. Next, we'll see next question, question number 8. When considered the dose response relationship for an essential substance, which of the following statement will come into? There are rarely negative effects of ingesting too much. No, this is rarely is the not the right statement. The curve is same for all people. No, it differs from person to person. Adverse response increase in severity with increasing or decreasing dosage outside of homeostatic range. If you see this particular graph, no, increasing concentration of toxicant with a constant exposure type on the x-axis, and response to the constant to the toxicant on the y-axis. Here see, this is the homeostatic region. Okay, adverse response increase in severity, increasing the dosage outside of homeostatic range. So that is the right option. Above this, in the blue region or purple region, you are seeing reversible damage and irreversible damage leading to death. Here only, once you cross the homeostasis, then it will cause. The relationship is linear. No, it is not. It can be nonlinear also. Deficiency will never cause more harm than over ingestion. No, this statement is also not right, since proteins, vitamins, hormones, all this deficiency will also cause some adverse effects, like vitamin deficiency, the night blindness, or those things will get. And over consumption of this vitamin A will lead to teratogenicity, fetal toxicity, reproductive toxicity features are there. So similarly like that. So what is the explanation is given is adverse responses generally increase in severity with dosages outside the homeostatic range. This graph is there, no? This particular is there, no? Both excessively high, excessively low doses. That is why we have to read about hormosis type of graphs, dose response curve, which are basically examples are hormones, your vitamins as well as minerals, right? Low doses can be harmful, while homeostasis is the body's state of stable internal condition. Our body will try to always try to maintain this balance, homeostatic balance. Exceeding this optimal range can disrupt the normal function. Whenever there is a disturbance in this homeostasis, then taking too much of a medication lead to toxicity. For example, acetaminophen. If you are taking more than 4 grams, that is 4 to 6 grams in a day, it will cause liver toxicity. Otherwise, it is good. Okay, taking a 500 or 650 mg dolo tablet, acetaminophen tablet 4 times in a day or 3 times in a day is safe. Shows antipyretic effect. But if you're exceeding it more than 4 grams of 4 to 6 grams per day, then it is causing liver toxicity. Included other example we have ethanol also. Lower consumption it is good, but higher consumption, repeated exposure and consuming in large quantities might lead to CNS consciousness, coma, all those things. While not taking enough might fail to treat a condition resulting in return or worsening of the symptoms. That's why hormones is curve and examples vitamins, minerals, and hormones, etc. Okay, just try to remember this type of this graph. Okay, so this is the threshold of safety. Okay, then we have question number 9. Question number 9, we have the therapeutic index of a drug. One question with respect to dose response curve, either with respect to hormones, they will give you different. When I wrote the exam, they gave different types of graphs, like shapes of graphs they have given and asked me to interpret that one in the option. So very important that one. So in this question, we'll read about therapeutic index, very well known concept, drug safety, descriptor, that is TI is the thing. What is this? Again, it's a revision like you read it. Is the amount of drug needed to cure or illness? No. The answer for this is efficacy. Efficacy is not ITI. Is lower in drugs that are relatively safer? No. TI is not related to safety. This is not the thing. TI is the comparison between the safety curve as well as the toxic curve as well as efficacy curve. Describes the potency of a chemical in eliciting a desired response. Potency is not directly TI. The potency, as we saw, based on at what dose it is causing toxic or adverse effect, that one low dose high toxicity, like that. Describe the ratio of the toxic dose to the therapeutic dose of a drug. This is important. This is the right answer. We need to know its ratio of TD50 by ED50. Okay, then explains the change in response to a drug as the dose is increased. That you will get it in dose response curve, not specifically related to TI. Okay, then let's see what is the definition. Therapeutic index of a drug is a crucial measure of its relative safety, and it's defined as a ratio comparing the concentration at which a drug becomes toxic to the concentration at which it is effective. That's why toxic dose is 50 and effective dose is 50 by 50. Okay, and you have to also know learn about the types of in the dose response curve, no, how steep is your dose response curve, that is also very important. This is the therapeutic effect, this is the toxin, and the distance between these two dose responses curves, no, that is your margin of safety actually. Okay, drug is taken, concentration amount of drug taken is the x-axis and percentage of response taken on the y-axis. So we'll see what these two inferences. Higher therapeutic index, if a drug chemical A is there or drug X is there and it is having high therapeutic index, for example 50, TI is example 50, means greater safety. These two distance, no, it is wider, then it is good. A drug with high therapeutic index has large margin of safety, MOS, meaning the effective dose is much lower than the toxic dose, right? Toxic dose is around 500 mg and your effective dose is 5 mg or 10 mg. There is a wide gap between these two doses. This makes the drug safe for the general use with less need for intensive monitoring. So in such cases, you need not monitor the drug concentration either in your body, in the plasma, like that. Example is penicillin, which has a very high therapeutic index. Okay, and well-known example, you can say acetaminophen. Acetaminophen efficacy dose is 500 or 650 mg, but the liver toxic dose is around 4 to 6 grams, 4,000 to 6,000 mg per kg, right? The next is lower therapeutic index means narrows the safety margin. A low or narrow therapeutic, whatever the dose response curve, they are like this, they're closer, means the toxic and effective dose are close together. These drugs often called narrow therapeutic index drugs. Examples for that is digoxin, lithium. Lithium is their anti-maniac drug, and calcium channel blocker, that's digoxin. Okay, anti-arrhythmic drug you can say. require careful, and valproic acid, phenytoin also have a narrow therapeutic index. Other examples require careful dosage titration and close patient monitoring. That's why these drugs, they need to be administered to the patient only in the presence of a well established physician. And see, individual patients should not take these drugs, often including regular blood tests to measure the drug levels to avoid toxicity or treatment failure. That's why digoxin also is always administered in the presence of the physician in the hospital setup only. So that was the important thing about TI. One question they will definitely, they try to confuse you with ED1, ED99, EC50, like that they will give all the options nearby. Then you will be, even though you might know the answer, but you will be not able to identify the answer at that particular time of exam. Last question is about penicillin. Penicillin interferes with the formation of peptidoglycan crosslinks in bacterial cell walls, thus weakening the cell wall, eventually causing osmotic death of the bacteria. Which of the following is true? So basically, from this question, we are trying to understand about the penicillin, how it is causing how the mechanism of action of this penicillin is there. Remember one thing. If you look into the cell wall, no, prokaryotes are different, eukaryotes are different. We are human beings, we are mammalian cell, our cell type is eukaryotic cell type. The cell membrane, whatever a phospholipid bilayer, no, it is different from that of the bacterial cell wall. So there is, whenever you administer penicillin in your body, that penicillin will not will able to identify the difference between the prokaryotic cell membrane and eukaryotic cell membrane. So it will selectively act only on the bacterial cell wall, not on the human cell wall. So we will see the concept here. Treatment with penicillin is a good example of selective toxicity. So that is the right answer. Other options we'll see. Penicillin interferes with the human plasma membrane structure. No, that's not. Penicillin is a good example of drug with low therapeutic index. Uh, in the previous one, we have seen no high therapeutic index. So this is not the right answer. Penicillin is also effective in treating viral infections. No, but particularly it is selective to the bacterial cell wall. That's why it is a bacterial infection only. Penicillin is completely harmless to humans. See, this statement is also not right because there are certain individuals who are sensitive to the penicillin. Okay, and that's why they might show some hypersensitive type 1 type of hypersensitive reaction, I guess, because of which any class of the same penicillin or beta lactam antibiotics, if the person is exposed for the second time, he will be having sensitive reaction, hypersensitive reaction. So this statement is also not applicable in this case. So right answer will be the green text, that is treatment with penicillin is a good example of selective toxicity. So it will target components of the bacterial cells, peptidoglycan cell wall synthesis enzyme, because of which the bacterial cell, no, it is unable to divide. It is not able to make the cell membrane. Okay, that is absent in the human cells. Can kill bacteria without causing significant harm to the host, making it a powerful and effective antimicrobial agent. So this is very important. Whenever you are developing a new drug, more specific selective should be there, so that other part of the human cells should not be get damaged. Okay, so hope from this video you will able to learn next 6 to 10 questions which were given in the Clayson textbook. Okay, um, we have learned a few concepts from this about therapeutic index. We learned about the selective toxicity. We learned about potentiation, additive, synergistic effect, that concept. Okay, so thank you so much. Be curious. For any questions, you can write mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel Toxicology on board. All the best for your exam.